Good morning and welcome everyone. My name is Kristen Burnham Johnson and I'm joined by my fellow science lead, John Barger. So John and I will each speak for about 10 minutes for our brief introduction this morning. Therefore, if you have any questions, please put them in the chat and we'll answer them there. So we're excited to discuss with you today the Environmental Molecular Sciences Laboratory, or EMSL's new directions, especially our interest in critical minerals and materials. But first, I want to take some time in my presentation to introduce all of you to EMSL and explain how our new community science campaigns are designed to address big scientific challenges. So for those of you who may be new to EMSL, we are a Department of Energy Office of Science user facility that's located at the Pacific Northwest National Lab, or PNNL. And as a user facility, we provide scientists with access to over 150 state-of-the-art instruments and the specialized expertise of our staff. So our proposal calls, they include FICUS, which enables access to multiple user facilities under one proposal, exploratory research, which employs certain highlighted capabilities that are actually developed at EMSL. And now we also have our community science campaigns that build on EMSL's capabilities to address large-scale scientific challenges such as critical mineral research. We're all very disappointed that we could not host everyone here in person for this meeting, but to give everyone a chance to explore EMSL, you can actually use this QR code on the slide for a virtual tour of the EMSL building. So the virtual tour is fantastic. Our comms team put it all together. It has 360-degree photos of research spaces and videos of researchers discussing EMSL capabilities. There's traditional videos embedded throughout the tour demonstrating research and or capabilities in action. We have photos and descriptive text that actually point the viewers to the EMSL website. And the virtual tour is essentially the most comprehensive way to see what we do without setting foot in the building. So please, I encourage all of you to go ahead and check out our virtual tour. Now, the tour is great because it gives you all a chance to learn more about specialized instruments and hear directly from our experts to understand what makes EMSL a unique scientific facility. As highlighted on this slide, so EMSL is equipped with world-class tools in chemical imaging, cell-free expression systems, and advanced analytical technologies alongside state-of-the-art computing, so AI, machine learning, data processing, and data visualization capabilities to turn all of our data into breakthrough discoveries. So researchers can access advanced molecular phenotyping technologies like proteomics, metabolomics, single-cell transcriptomics, optical imaging, NMR, and advanced tools for structural biology. Plus, EMSL has unique terraform synthetic habitats, which enable multidisciplinary innovation and discovery. Now, this meeting, so both John and I are hosting this introduction because it's associated with our science areas. So this meeting is hosted by the Functional and Systems Biology, or FSB, and Environmental Transformation and Interactions, or ETI, science areas. So with FSB, we focus on uncovering biochemical pathways that connect gene functions to phenotypic responses. We study molecular interactions within microbial cells, within microbial communities, and their environment. And so the key research platforms for FSB include structural biology, biomolecular pathways, and cell signaling. And John will talk more about this in detail in his presentation, but ETI, it explores the environmental and ecological processes to create an abundance of data, and this abundance of data can inform large-scale models. So together, these FSB and ETI areas, they connect biology and environmental dynamics to address complex scientific challenges. And most importantly, our campaigns, which we're very excited about, they're tied to our science areas. And our campaigns feature greater community input and focus scopes to support big science achievements. So that's what we're doing today and tomorrow as part of this meeting. So many of you will be part of the breakout sessions today and tomorrow to help provide us input and focus the scope of 3 of our campaigns. So for ETI, there'll be 2 breakout sessions for 2 of its campaigns. The first one being rhizocritical, so critical minerals biogeochemistry in the rhizosphere. And the second one is unconventional critical, So biogeochemical processing governing critical minerals recovery in mineral-dominated systems. Now for FSB, there'll be one breakout, and it's associated with our campaign of metal-binding biomolecules, so discovery and characterization of critical metal-binding small molecules and proteins. Now, using FSB as an example, on the next few slides, I want to emphasize that the goal of our science areas is to address DOE, biological environmental research, or BER research goals and grand challenges. So for FSB, we're advancing systems biology to understand and manipulate microbial genomes and developing predictive knowledge of nutrient, metal, and elemental cycling in complex systems. And what I'm going to discuss on the following slide is FSB campaigns and how these campaigns are helping us prioritize these goals. So most importantly, EMSL is now more nimble through campaigns. So these are our 4 FSB campaigns. They're live on our EMSL website. And these campaigns, they will help our users by leading to improved strategies for designing fungi, bacteria, and other microbes, for the production of bioproducts, biofuels, and more to help our bioeconomy, as well as unraveling the complexities of nutrient, critical metals, and rare earth elements cycling within microbes and their immediate environment. So these campaigns, they're built for our users. They're built for you. So to make engagement easy, there's a lead scientist for each campaign. And campaigns, they enable big science by helping groups of users come together to overcome big challenges in the field. And this is my last slide and my most important slide. So the FSB campaign that we're going to be focusing on today is Will Q's metal binding biomolecules campaign. And our goal is to help DOE, BER funded and aligned researchers tackle the challenges pictured in the middle of this slide. So how do we take dilute rare earth element sources and transform them into enriched CMMs? Well, we first understand and manipulate microbial genomes so we can use them for the bioprocessing. And most importantly, how can EMSL instruments and capabilities help? Well, EMSL can help with the 3 bullet points on this slide. So EMSL can help with bullet point 1. EMSL can help identify enzymes and metabolic pathways in microbes for FSB and plants for ETI that require critical minerals and leverage these mechanisms for extraction and recovery. EMSL can help with bullet point number 2, identifying novel biomineral forming biomolecules, pathways, and regulatory networks. EMSL can help with bullet point number 3, so engineering biological systems to concentrate and produce critical biominerals using modified microbes. And for all of you participating in our breakout sessions today and tomorrow, you will help us design and structure this collaborative campaign. And with that, it's my pleasure to pass it over to John.
I'm John Barger. I'm the science area lead for the ETI, or Environmental Transformations and Interaction Science area. And I'm just going to pick up where Kristen left off. So the ETI science area is focused on molecular and microscale processes that control metal, nutrient, and element cycling in chemically and physically complex soil and subsurface systems. This is an important goal of the BER, Earth and Environmental System Sciences program. Another major goal, shown on the right, is advancing predictability of the Earth system at the largest scales. ETI is supporting this goal by standardizing biogeochemical workflows and generating high-quality data that can be directly compared and combined across the continental United States. Our campaigns are designed to target both of these important goals. So speaking of campaigns, I'd just like to emphasize what Kristen said, which is that these community campaigns will elevate the science impact far beyond what an individual PI could achieve in a smaller project. By enabling and supporting interactions between leading researchers, we can tackle truly large-scale science challenges. And while this is super important for BER, this also creates terrific opportunities for you. So we're really glad to see so many people participating today. ETI, as Kristen mentioned, is supporting 2 critical minerals-focused campaigns. The Rhizocritical campaign leverages EMSL's deep expertise and capabilities in understanding microbial function of rhizosphere systems. The target mineral is nickel, which is enriched in ultramafic soils. There's great interest in using nickel hyperaccumulating plants to extract nickel from these soils because of the large size of the soil's resource. But the microbial communities and how they're interacting with those plants are critical to understand. To this end, ARPA-E has funded several phytomining projects, which we hope to leverage and amplify in this campaign. The unconventional critical campaign, led by Odetta Chifaku on the right, I should say the rhizocritical is led by Amir Akami. You'll get the chance to meet all these folks very soon. The unconventional critical campaign is focused on other unconventional sources of critical minerals, including mine tailings and mine waste outflows, shales, weathered laterite, regolith, etc., which contain, for example, cobalt, nickel, and rare earths, but are too dilute for conventional recovery methods. In situ biogeochemical leaching methods in heaps or below ground in situ recovery are of great interest here, as well as many, many other technologies we'll be discussing today, because of their lower energy and overall costs. However, there are many fundamental challenges to be addressed, including identifying the chemical and physical forms of the critical minerals and materials in these earth materials, the biological and metabolic processes that solubilize them, and manipulating the rates of these processes within the constraints present in these complex multi-phase systems. So ETI campaigns are focused on these so-called upstream processes involving mineral microbe solution interactions because hydrobiogeochemical processes, such as these illustrated here, fundamentally govern critical mineral and material recovery from soils and sediments. Every discovery in this fundamental space will open new opportunities and be multiplied many times across applied R&D efforts. This is why these campaigns are so important. ETI has deep experience resolving biogeochemical processes in complex pore-scale environments. And as many of you know, resolving such biogeochemical interactions often requires spatially resolving them over a range of scales from nanometers to millimeters, as well as providing spectroscopic economic measurements and often complementary flow and transport modeling. To this end, EMSL has developed several advanced capabilities to address exactly these needs, and we're leveraging them in the campaigns. These include, on the upper left, micromodels to allow manipulation of sediment pore structures and flow and image spatially resolved processes in situ. Lower left corner, microscopy and bioimaging capabilities to co-locate and chemically identify metals, organic cells, metabolites, and proteins. Upper right-hand corner, chemical analytics provide deep data on organics, nanomineral phases, and omics, all of which are important in this realm. And last but certainly not least, lower right-hand corner, rich standardized molecular data from field samples across the continental United States help us to understand and model important biogeochemical processes across sites and regions. Speaking of the latter point, one of our premier capabilities is the Molecular Observation Network, which I'll refer to as MONET, which collects and processes field samples through standardized workflows to generate advanced gold standard molecular data for user research and for Earth system scale modeling. Campaign participants submit standardized samples, and EMSL performs predefined suites of measurements using premier capabilities shown here on the right side via standardized workflows. The data is feature-rich. For example, there are thousands of peaks in each soil organic carbon ICR mass spectrum. You can see it here in the figure. You can see that there are thousands of peaks. This is very important for AI modeling. The data is indeed very well suited for AI applications and has deep predictive value because of this richness of features, as well as the fundamental molecular character that encodes biological and geochemical function into the data. Moreover, the ability to compare samples and processes in an apples-to-apples fashion across regions is critical to enabling deep understanding of processes for model development. The metagenomes that we're developing are a resource for mining biomolecule functions, as Kristen noted. And most importantly, perhaps, data from ETI campaigns is being released as structured AI-compatible gold standard data in the MONET database. These data are fully open and available to all potential users. So I'd like to shift to the value proposition to you all because it's huge. I just want to emphasize the campaigns do offer tremendous value to you and the user community. Students and scientists that submit samples get advanced standardized suites of molecular and microstructural data for their research. This can be vital to early careers. We provide extensive training, which I'll discuss in the next slide, and the growing community is an important source of networking, learning, and reviewers for your work. I already mentioned that we have standardized data also from across the continental United States. EMSL training and outreach provides numerous opportunities for students, postdocs, and scientists. We have training at annual community science meetings, and this year we'll also feature this at the 2026 EMSL Summer School for building skills, networks, and relationships. We also have monthly working groups that provide guidance for interpreting data and writing papers on a continuing basis. So the concepts that you learn in these less frequent annual training meetings can stick and become actionable for you, even if you're not an expert in these methods. We're hosting sessions at national meetings to provide opportunities for you to network and share your science. And lastly, anyone can participate in the campaign proposal calls. So this is my last slide. What will we do today and tomorrow? The goal of the meeting is to identify science knowledge gaps and technical challenges hindering our ability to improve critical mineral recovery from unconventional sources, and then jointly ideate multi-investigator user projects that address important grand challenges in each of our campaign science areas. As illustrated here, we'll start with keynote talks to set the stage, and then proceed through a series of focused breakout sessions intended to generate campaign project concepts, and these will be led by our three campaign leads here shown on the right, Will Q, Amir Akami, and Odetta Chifaku for the metal binding biomolecules campaign, the rhizocritical campaign, and the unconventional critical campaign. Our ask is that you please tune out your busy office environment and participate as you would have done if you were here in person. And with that, I'll say thank you and hand the baton to Odetta Chifaku, lead for our unconventional critical campaign.
Well, thank you, John. I am Odetta Chifaku, as John mentioned, and I'm a soil geochemist with EMSL, and I'm leading the EMSL Unconventional Critical Minerals campaign. And I'll also be facilitating the breakout sessions on this campaign later today and tomorrow. Now it's my pleasure to introduce the first keynote speaker of our plenary session, Dr. Heileen Hsu-Kim. Dr. Hsu-Kim is a professor of environmental engineering at Duke University. She earned her bachelor's degree from MIT and her Master of Science and PhD from UC Berkeley. Dr. Hsu-Kim is a leading expert in aquatic geochemistry, trace element biogeochemistry, and nanogeoscience. Her research explores innovative ways to recover critical metals, such as rare earth elements, from coal mine waste and other legacy materials. She also serves as Director of Graduate Studies in Duke's Civil and Environmental Engineering Department. Please join me in welcoming Dr. Heileen Hsu-Kim.
Great, thank you. Thank you, Odetta, for that nice introduction. I really appreciate the invitation to speak here and to talk about our work with this great audience here. I'm looking at the attendees list and really so glad that to have you here. So what I'm going to be talking about to set the stage here is examples of our work looking at rare earth element resources from waste streams. In this case, we're focused on coal-based waste streams. And I'll talk mostly about our work related to acid mine drainage. This is work that is done in partnership with Anushka Mishra, who's a postdoc in my lab, as well as Ben Hedden, who's an environmental scientist with Hedden Environmental. So our work is focused on rare earth elements as waste streams, as I mentioned before. And some of the big science questions that have come out of some of this work that I want to kind of set the stage for is that our work kind of really illustrates, or at least highlights, the fact that when we're trying to resource waste streams for critical metals, typically we're talking about feedstocks that are really spatially dispersed, so geographically dispersed. So for example, we're talking about mine drainage here, as well as coal combustion residuals, which I'll touch on real briefly. Not only that, this waste stream is quite complex in terms of the chemical composition and diverse, and so that makes it a challenge in terms of developing recovery processes. And then at the very end, I'll talk about some examples of what that means for extraction. So, why acid mine drainage as a resource? Well, in northern Appalachia, there's been a large number of studies that have identified acid mine drainage from abandoned coal mines as being enriched in rare earth elements. And these would be the drainage sources that are the most acidic. So we're talking about those that are pH values less than 3, less than 4, that will have milligrams per liters of rare earth elements dissolved in the water. And if you take the hundreds of sites that are located all across the northern Appalachian region and extrapolate these concentrations to the most acidic sites, as well as that they're producing this water continuously without an end, this resource adds up to about a couple thousand tons of rare earth elements per year, just from these acid mine drainage sites. And this adds up to about the production of a small rare earth mine. In this case, I just also want to highlight that rare earth elements I'm defining here as those elements in the lanthanide series, as well as yttrium and scandium. In terms of the how we deal with these acid mine drainage, so this drainage is obviously has a negative impact in terms of local water quality and ecosystems, and so these drainage sites need to be treated. There's various strategies that are involved in treating acid mine drainage. They can involve active treatment, so we're talking about diverting these flows into systems where we're basically doing chemical neutralization. And then there's also passive treatment systems. These are kind of more nature-based solutions where you can develop you can divert this water into a series of settling ponds to help oxidize that water, settle out the metals, as well as promote acid neutralization. Other systems that I'll focus on in terms of our work are other types of systems that include drainable limestone beds, which is shown on the bottom here, which in this case, the water is diverted into these limestone beds that neutralize the water. And then those beds are periodically drained, resulting in the release of flocks that developed in these limestone beds into these settling ponds. Another strategy would be to divert this water into wetlands in a vertical flow format. So these wetlands are constructed with material that includes limestone and then layers of organic matter. The idea here is that you can neutralize the acidity in this water. The water flows vertically from top to bottom. It neutralizes acidity while also keeping it anaerobic to prevent the iron from flocculating out and clogging all this. So what I'll talk about here is our work sampling from these two kinds of systems in the northern Appalachian region. Some other things that you can see just from some initial data is that, in some of the systems that we've sampled from, the rare earth element concentrations are in the hundreds to 1,000 micrograms per liter. And then the respective rare earth element concentrations in the solids that are produced from these passive treatment systems are then enriched. So we're talking about several hundred to more than 1,000 milligrams per kilogram of rare earth elements. And just to really highlight that these systems are generally in rural mountain locations. So, excuse me, we're talking about remote locations. These are just examples of flush ponds and settling ponds and vertical flow ponds that we've sampled from. And the systems would be kind of in, well, I meant to here to just highlight that these are geographically in places that are not necessarily centralized. So that's an issue when we're thinking about recovering critical metals from these systems. I want to throw in here just one slide on coal ash. Coal ash are the residuals produced when we burn coal for electricity, and has also been proposed or is being looked at as a resource for critical metals like rare earth elements. And so on the graph on the right is just an example of data that we published a few years ago on rare earth element concentrations for different types of coal fly ash. And the concentrations really depend on the type of coal or the regional source of coal that was burned to produce that ash. And there's a moderate level of enrichment there. I think what makes this resource, unconventional resource, attractive is the fact that we've been burning coal for many decades, and a lot of this ash has been disposed of at landfill sites right next to power plants that produce this ash. So the map on the left is showing locations of current disposal units at all the major coal-fired power plants in the United States. And a lot of these power plants are not burning coal anymore, but these sites will contain millions of tons of ash per site. And if you add them up, in terms of the amount of coal that we've been burning over the last 2 or 3 decades, the discarded ash adds up to about 2 billion tons of material. And not only that, the recent rules by the EPA are actually going to require a lot of these coal ash ponds to be retrofitted and closed, meaning the material needs to be excavated and moved into lined landfills because of the problems with this storage. So this is just another potential resource, unconventional resource. But let me go back to acid mine drainage. I think one of the challenges, so this is my second point, is one of the challenges with acid mine drainage for resourcing is just the complexity as well as the diversity of the resources of this resource. So the graph on the right is showing some data of drainage feedstock, acidic feedstock that we've collected from several different sites in southwestern Pennsylvania. And I'm plotting here the concentrations of selected rare earth elements in green, as well as other critical metals like lithium, cobalt, and nickel. And you can see we're talking about hundreds of micrograms per liter. But also what's in these fluids are metals like iron, aluminum, manganese. That is 100 to thousands of times more concentrated, as well as sulfate. And so we have, in terms of the extraction, the challenge of getting the metals that we want is actually the fact that we need extraction methods that can handle the selectivity that we need, the 100 to 10,000-fold selectivity that we need. And not only that, these sites are geographically dispersed, so an extraction process or a refining process will need to be able to deal with the wide range of scale in terms of implementation. So the modularity of the unit process is really important. Here I'm showing just from the same set of samples, just the balance of these major metal ions that vary between these different mine sites. And here I'm focused here on iron, aluminum, and manganese. And this is the feedstock. This is the acid mine drainage water. And what we observed is that some sites seem to be really high in aluminum concentration relative to these other metals, while other sites tend to be what we're calling iron rich, just have really high levels of iron relative to the other metals. One site being manganese rich. So this is what I mean by the diversity of the feedstock. So not only is it complex, but there's also just a wide variety of what I mean by complexity. And this actually really impacts, this is really important in terms of sequestering the metals that we want, which is like the rare earth elements, because that will actually, this balance here will likely dictate the host mineral phases, the co-enrichment of what might be extracted, co-extracted when we're trying to get rare earth elements and other critical metals. So here's an example of actual data of what I mean by that, how feed water chemistry can actually impact the enrichment. So on the graph on the left is the same data that I showed before, showing the rare earth element concentrations in the feedstock, the rare earth element concentrations in the solids that are precipitating out in these passive treatment systems. And then this is actually color-coded according to whether we're talking about an iron-rich or aluminum-rich or manganese-rich feedstock. The graph on the right is showing the same data that's plotted on the left, but now plotting it as a partition ratio, defined as the concentration in the solids over the concentrations in the influent. And you can see that a lot of the orange ones, the orange ones, which are the iron-rich influent, tends to have a lower partitioning. The blue one has a higher, the blue, which is the aluminum rich, tends to be higher. This one manganese rich tends to have a really high partition ratio. And there's a lot of things that explain this. One is that we think these orange ones, the iron rich, the reason for the quote low partition ratio is because there's just a lot of iron in the water that is co-precipitating with the rare earth elements and basically diluting it in the solids. And we don't get that in some of these other aluminum rich feedstocks. So this is where the feedstock composition of the metals we don't want or actually could be influencing those that we might be targeting, like the rare earth elements. The other question is, given that rare earth elements are concentrating in these solids that are precipitating in these treatment systems, what are the chemical modes of association of these rare earth elements? And so here's some SEM, initial work that we've done using SEM and element mapping with EDS, showing kind of giving an idea of like the grain size and morphology of these precipitates. The story here is basically we have a big mess of materials. It's really heterogeneous, a large variety of potential host phases. So for example, this group of images on the top is example of solids that were produced in this manganese-rich influence system. And so there's a lot of manganese precipitating out in the residuals, a lot of aluminum as well. And you can kind of see here just from the mapping that some of these particles are rich in manganese and aluminum. Some of them are rich only in aluminum. There's also iron present, for example. And, I think there's a really great opportunity to dig deeper as we then use other methodologies to tackle the associations of the critical metals like cobalt as well as rare earth elements, and try to examine how there might be different between these two, and that would be important in terms of the how we then extract the rare earth elements from these solids when we harvest these solids from the treatment systems. On the bottom, you'll see an example with a site in which the water is aluminum rich, and the residuals are even more aluminum rich relative to iron and manganese. And so we have a lot of the material here has a lot of aluminum containing particles, presumably aluminum hydroxides. So kind of the other point here is that there's co-enrichment of aluminum in the solids. And then, what we haven't touched on at all is that in these systems, there are microorganisms in these systems that's probably catalyzing, say, the oxidation of manganese, for example, and just being involved in these precipitation processes. So that's definitely an open question that I think our community is really well poised to address. I want to touch on this other treatment system that is a vertical flow pond that I think is also really interesting. So this is a type of passive treatment system in which the water is diverted into a system that is like a vertically flowing system. These are usually filled with limestone and some kind of organic matter. And this one at Jennings was filled with a compost, like a mushroom compost. And these systems are usually in operation on a scale of like 10 to 15 years and then need to be, I guess, reconstructed. So it's like in those like 10 to 15 year time scales in which this material would be, after it would be reconstructed, and that this whole thing would be excavated out and refilled. So it's in that excavation time in which you can actually have this material that can then be a resource to harvest for critical metals extraction. In this case, our team was actually interested in understanding if there's vertical stratification of metals in the system. So this material is probably a couple meters deep, and it fills in over time. And what our team tried to do was collect cores. So you can kind of see Ben and my former student Anna collecting cores and trying to just get an idea of some stratified layers from this system. And so what I'll show you here is some data of what we observed in terms of stratification of rare earth elements. We observed, so this is plotting data in one of these cores. Water is going from the top to the bottom, sort of reiterate. And what we observed is that the rare earth elements seem to be accumulating like not at the surface, but right below the surface. in this system. So you get this for these 3 rare earth elements. You can see the peak of the concentration right below the surface. I have other data I want to show you. Just same core. Just highlighting, what else is stratifying in this wetland system. So along with the rare earth elements, aluminum appears to be concentrating right below this, right below the surface in the same area that the rare earth elements are accumulating. And keep in mind that as the water goes from top to bottom, it's getting neutralized in terms of the acidity because of the limestone that's present here. So the pH is certainly changing from the top to the bottom. And when it comes out, at the bottom into the effluent, it will be neutralized in terms of pH value. What's also interesting is that we observe that the transition metals are accumulating in this wetland, but at much deeper depths. So cobalt and manganese is what I have as examples of transition metals accumulating kind of at a different spot than where the rare earth elements are accumulating. And my suspicion is that the cobalt which is at trace levels relative to some of the other metals, is probably being co-hosted with manganese oxides and hydroxides and iron hydroxides. So that would be important in terms of when you're trying to extract it, understanding like how these metals like cobalt and rare earth elements are associated, so that we can then design the extraction process. Oh, and then I forgot I had one more here, is that some SEM images of what this material looks like. So this is the top is a sample of the near surface. So this is the depth interval where rare earth elements are elevated, and we can see that there's a lot of aluminum containing material, and as well as silicon. And the iron is just kind of diffuse. It's not as strong in terms of I guess concentrating on the specific particles. While as in this deeper layer, so this is the layer where cobalt and manganese was really accumulating really strongly. You can kind of see the signals of the manganese and iron and aluminum being really strong here, whereas there's less in terms of the silicon. So I think this really highlights that there's really a wide variety of host mineral phases for the target critical metals like the rare earth elements and cobalt and nickel and all that. So in my last bit, I want to talk about extraction processes. So there's a large variety of extraction processes that you can implement when we're talking about concentrating rare earth elements from waste streams. Typically, the goal is, we take some kind of acidic feedstock. So this could be acid mine drainage. It could be the leachate of coal ash. It could be the leachate of some of these treatment solids. And we put it through a bunch of different steps to then come up with a mixed rare earth product that can be saleable, ore grade, further separated to neodymium or the individual metals that we want that can then be used to make magnets and things like that. When we're talking about geographically dispersed sites, so like acid mine drainage, I think what is probably the most feasible in terms of getting from this feedstock to the mixed product is actually to implement some kind of in-between steps on site or near the site that then you take the mixed product and then transport it to a more centralized site for further processing. And so there's different approaches that you can take to do that first initial extraction. The key here is that these processes need to be modular. They need to be able to handle a wide variety of flow rates if we're doing this on site. So what I wanted to show you here is an example of, say, doing pH adjustment. So this is what's done at the pilot plant that is run out of the University of West Virginia, where they're taking acid mine drainage and actually doing an initial, basically selective precipitation by titrating the pH to higher values to selectively precipitate out residuals. And in this process, so I'm calling it staged precipitation. Maybe it's called something else at this pilot facility. But we're taking basically acid mine drainage, so it'd be really low pH, and then at certain target pH intervals, you can actually precipitate out certain solids, and the rare earth elements would then be concentrated at probably around pH 5, maybe pH 6 is usually the target that is done. So here I'm showing you, this is not real acid mine drainage, by the way. What I'm going to show you here is some data of a simulated acid mine drainage, where what we did was we made water based off of the composition of one of the field sites that we went to, one of our iron-rich field sites, where we have iron, the key constituents, iron, aluminum, manganese, the lanthanides for sure, cobalt, some of the transition metals as well, and then also a lot of sulfate here, a lot of sulfuric acid. And did this kind of stage precipitation and collected, you can see the filters here, collected solids from this. And, these solids in terms of their composition are actually really quite variable. And I think the solids that are produced, or at least the chemistry here that's happening, there's a lot of opportunities for our community to really dig in and understand what is being precipitated and why. So this is an example of one of these, what I call pH titrations. Is like, you can kind of see as you increase the pH, we get a loss from solution of our major metals, aluminum, iron, and manganese. The loss from solution occurs at different pH values. And then, rare earth elements being removed from solution at this pH, cobalt at a higher pH. And then what that means in terms of their concentrations in the solids is that you have high rare earth element concentrations at the pH 6 to 7 precipitation points, cobalt being enriched at the high pH points. And then when you look at what the major elements are, where their concentration, this is a stacked bar chart of the three that we're really focused on. And you can see that the rare earth elements, which are the pH 6, 7 points, these solids are rich in aluminum and iron, whereas the cobalt containing samples would have a lot of manganese in it. So this would be really important, be really interesting. Well, so we need to do this with real acid mine drainage, but it'd be really interesting to then characterize the solids here. I think another thing and I didn't have this, but another thing here is that, iron is also being oxidized in this process. So, I don't want to say that this is all iron, ferrous iron. There's definitely ferric iron here. And then also with acid mine drainage, you have microorganisms that actually catalyze the oxidation of iron. So iron, if you have, instead of ferrous iron, you had ferric iron in so iron plus 3, this titration will look very different because iron 3 will precipitate much quicker at lower pH values. And that will then impact the whole process. And so, when we do this at scale at a real processing facility, the oxidation of iron is really important. The role of microorganisms will definitely play a role here. And then I know I'm running out of time. I just want to touch on other extraction methods for the rare earth elements specifically. Our lab has done a lot of work with supported liquid membranes. This is a liquid-liquid extraction approach for rare earth elements, for metals in general, in which we're using the conventional solvent extraction type approach that's used for rare earth element refining. The key difference here is that our organic solvent, which contains our extractant, our metal chelating ligand, is actually embedded in a hydrophobic membrane. And so that when you operate the system, you have your feedstock on the left, so our acid mine drainage, you have your stripping solution on the right, and the organic layer in the middle. And through metal proton exchange reactions that take advantage of the ion gradient across this membrane, we get selectivity for rare earth elements going from the feedstock into the stripping solution. And the reason for doing a membrane-based method is that you can maximize the interfacial area between all these different phases. And then as a membrane, you can actually implement this as a modular process design. But what I want to illustrate is that when we're dealing with then unconventional resources like acid mine drainage, the efficacy of this kind of membrane-based system, the flux, is really guided by, at least is really impacted by the water quality of the feedstock. So here I'm showing some data showing permeability of neodymium. Permeability is the normalized flux of neodymium across this membrane. And you can kind of see that the permeability will actually increase for greater pH values of the feedstock. So this is due to at higher pH, you have a greater proton gradient between your feedstock and your stripping solution. And then you also see here on the graph on the right that the metals concentration, the major metals, particularly aluminum, iron, manganese, and calcium thrown in here, the higher concentration of these competitive cations, the lower the flux. So that's what this graph is showing. So the key here is that there's a lot happening when we're dealing with complex, relatively dilute feedstocks in a liquid-liquid extraction process in which you have metal competition, you have proton competition, all at these liquid-liquid interfaces. And I think as a community, we've done a lot of work on, interfacial reactions of trace elements in complex media. And I think with liquid-liquid interfaces, I think could be really interesting thing for us to look at. So this is my last slide. I just have some main bullet points of what I think are, well, they're summary points, but what I think might be really good research opportunities. One is that if we're looking at unconventional resources for critical metals, we should really focus in, or at least consider, the major element composition. And the fact that some of these elements like aluminum, manganese, are probably these unknown host phases in which the critical metals are associated with. I also want to throw in that hydromedical allergical processing and the efficacy of it is really guided by interfacial processes. That's something that we can hone in on. And then microorganisms and mineral transformations are kind of interlinked. These are not abiotic systems for sure. And ideally, so this is like kind of my holy grail of what I don't know what to call it. If we can take some of this information that we gather at local and molecular scales in terms of the quality of the feedstock, the associations, things like that, and link that to geospatial data to then come up with like an inventory of the resources. I think that would be really cool to do. It's something that our group has done with coal ash. So this graph comes out of this map comes out of a paper that we done. We did that for coal ash for major elements. But I think it'd be really great to do that for mine waste, coal mine waste, other kinds of hard rock mine waste. All right, so I should stop and I'm happy to take any questions. Thank you.
So the first one that was posted here was, and this refers to the coal mine, data that you showed in the map. And somebody asked if you have any data from Puerto Rico in that one. So this is a quick question.
No data from Puerto Rico, unfortunately.
No data. Okay,
Yeah.
Perfect. So the second question is, what are the major sources of this high concentration of metals? Are they natural or human-led contamination? This is from Prakash.
Yeah, the places that we've gone to are coal mine drainage sites. So the coal seams are enriched, naturally enriched in rare earth elements. They're anthropogenic in the fact that these are abandoned or inactive coal mines that are continuously leaching from these sites. So that's the anthropogenic part, but the rare earth element occurrence is natural, like a geological formation.
Yeah, great. So the third question is from Ejiro, I think. Maybe I'm saying the name wrong. Given the complexity of how rare earth elements are associated with different host materials, how might these findings influence the downstream value chain for critical materials? And then there's a follow-up. For instance, are there supply chain adjustments we should consider to better manage more heterogeneous feedstocks?
Yeah, I think, how do we use this information in a useful way? Maybe that's ideally this is what I'm thinking, is that if we can get an idea of categories of types of feedstocks, because right now, like, we just know it's a lot of different types, complex, diverse. If we can somehow categorize it according to then what would it mean for the extraction technology, then that can help feed into things like, okay, like and like an techno-economic feasibility like assessment. Like, okay, how many sites do we need of this kind of quality to produce X amount of rare earth elements? Because I think it's the amount of rare earth element that you can produce given a collection of sites is really what's important. And that is what you need to then basically be able to feed information into understanding the supply chain aspect.
Great, thank you. So the next question comes from David Hoyt. He's a scientist here in EMSL. And he asked, in the simulated AMD experiment that you showed, did you think of amendment with microbial strains to aid the recovery of REEs?
No, that's a good idea.
Awesome, great.
My question would be, is that feasible to do at scale? Yeah, that would be my question. I'm sure it is. Yeah.
Okay, moving along. Chuan Hu he's thanking you for your great talk and is curious about the selectivity for the current approaches that you mentioned, like solvent extractions, ion exchange, and liquid membrane. Are they selective for the separation? I think he means selective to a specific rare earth.
So solvent extraction is what's used in conventional rare earth element mining. So yeah, the selectivity is definitely there. It's used at pilot facilities developed for extracting rare earth elements from geological waste streams. You have to have like multiple cascades, like many separation steps to get individual elements, like the neodymium separated from the dysprosium, for example. Did that answer the question, Odetta?
I think so. Yeah,
Right.
Yeah. So the next question it comes from Tomas Varga. He's also an EMSL scientist. He says, thinking about scaling up this process, how big of an issue is secondary waste, water, and solid residues production?
Yeah, big. I mean, we're taking something that is like, 1 in 1,000. So one part, what we want, 990 parts, what we don't want. So like, there's going to be the residuals is an issue. I think co-production is the realistic solution to dealing with any kind of unconventional waste stream. So for example, like coal ash, it's about you're taking the material. If you're extracting the critical metals that you want, you produce these residuals that ideally could be used for structural materials like concrete and cement. At acid mine drainage sites, there are real examples of using these solid residuals as a resource for specialty products, like especially iron-based products, for example, pigments. Magnesium, I think, could also have resources. So there's definitely the co-production is definitely how these are going to have to be implemented. In terms of the other kinds of environmental impact, I think what I really worry about is that a lot of these processes, if they require some kind of leaching step, will then mean that they'll consume chemicals like acids. So the approach of like bioleaching, I think, is, for me, attractive because of that. And I know there's a lot of people in the audience who are experts in that. So I think there's a lot of moving parts here, but yeah, definitely thinking about the holistically of like, what are the trade-offs and what do we have to think about in terms of co-production, I think is really important.
Great, thank you. So the next question, I think it was from John Charover, and he mentions, according to slide 18, which I don't remember right now, which one was that? Solid phase products of stage AMD neutralization contain about 100-fold higher iron or aluminum phases hosting REs. Are there established approaches for selective extraction and enrichment of REs from these precipitates? I think this goes back to the selective approach that you mentioned.
I mean, the thing is with these precipitates, so we put these precipitates on the XRD and we weren't getting any peaks. So these are all amorphous. It would imply an ease of leachability that you wouldn't have out of like waste rock material, for example. So I would expect that, like, for example, if you need to leach these out, you could probably get away with more dilute acids, less hazardous acids as a way of extracting. Hopefully that answered your question, John.
Okay, and then this is the last question, and then the rest will pass it to you so you can answer them. So you mentioned that critical minerals come from diverse regions. Are there biogeochemical processes that might be common across these regions from what you saw? And then could identifying those processes help generate data or model predictions that are transferable across other sites?
Yeah, how to make this
Yeah,
like more than just this site, this site, this site.
exactly. Yeah.
That's a question that hangs on me all the time as like a black cloud. I think maybe an understanding of why, like, for one is like there's a lot of patterns that kind of develop, like in instances in which we see aluminum hydroxides or aluminum coming out of solution, presumably forming hydroxides. This is in a select stage precipitation experiment. This is actually in these treatment systems in our vertical flow pond. Rare earth elements are also coming out of solution. And so I don't know if aluminum hydroxides are the host phases, but like I would presume that they are. Why is that? Can we develop geochemical models for that? I think it's mostly chemical, but these are not abiotic systems. So I think that's like one example that I can think of right away. But I think there are others to discover as well. I think manganese is also something that we could take what we already know about manganese oxidation.
Okay, great. Thank you again for a wonderful presentation and a lot of questions that came your way. And so next presenter will be introduced by Will Q.
Thank you for that, Odetta. And thank you, Helen. That was a fantastic way to start today. I certainly learned a lot. So next up, we have Rene Boiteau. He is a assistant professor at the University of Minnesota and previously actually was a Linus Pauling Fellow at PNNL. And it's really exciting to introduce him today to talk about mining environmental microbiomes for selective metallophores. So Rene, if you want to take it away.
Well, thanks for organizing such a really great workshop. I'm super excited to learn more about critical minerals from unconventional sources. So today I'm going to talk about some of our work developing analytical tools to actually probe some of the mechanisms that microbes and plants in the environment use to mine the elements that they need to grow. And so this is focused on a slightly different suite of elements than the previous talk. I love this periodic table shown on this slide that shows the elements that are required for life. And the point here is that organisms need carbon and hydrogen and oxygen and macronutrients like nitrogen and phosphorus to grow, but they also need a whole host of other metals, and they have to somehow get these from their environments. In the case of these metals like first row transition metals, they're primarily in the environment in very poorly soluble mineral phases that have to be accessed somehow. And so biology has developed really effective strategies to pull these metals out of minerals in the environment. Right? So there's a lot of overlap between the metals that these organisms need and have learned how to mine, and the metals that are critical for green energy technologies, and that we need to somehow improve our mining strategies to meet growing demand. Right. And so the toolbox of biology for mining metals is actually the same as our toolbox for mining metals, right? We can have acidification where you just dissolve rocks through protonation that's hydrolyzing oxygen metal bonds. There are reductive processes. So that would be an example of a pretty unselective process that's dissolving minerals and mineral surfaces. There are reductive processes where electrons or electron donating organic molecules are released by microbes to be able to reduce elements to convert them into more soluble phases. Right. And then finally, there are chelation processes where specific organic molecules that have multidentate ligands can bind to metal on a surface of a mineral and pull that off because it can form a very, very strong complex. So those chelation strategies can be the most selective because the chelates can be tailored to have stronger binding for one metal over another. And with chelates, this is often designed to be catalytic. So if a microbe is releasing a chelator, oftentimes these are metallophores that are primarily produced with the sole purpose of mining a specific element. Right, it will dissolve that metal in the form of an organic complex that will be taken up back by the organism, and then there are particular enzymatic mechanisms to remove that metal, either through reduction or hydrolyzing a bond that can then be repaired, and then that chelate is recycled. Right, so this is sort of a catalytic process for improving metal dissolution rates. And in nature, all of these processes end up working together, and there's synergy between reduction and chelation. But as a selective approach, a lot of our work has been focused on, what are the pathways by which microbes are solubilizing through the production of specific organic chelators. And this becomes a very interesting challenge in the environment where we can have lots of different co-occurring organisms that have different strategies for chelating different metals and then facilitating their uptake. Right, so question that we focused on are different environments. How do plants and associated microbes control metal bioavailability? So all these different plants, bacteria, and fungi need copper, nickel, zinc, cobalt from the minerals in their environment. They can produce different organic ligands that can dissolve those metals, and then somehow those have to be taken up. So we can ask some very basic question: what compounds are binding to these trace elements in soils and other environments? Which metals do different chelators bind? And then which organisms can take up those specific elements? And this gets at synergistic effects between organisms that are mining metals and then other organisms that may be using them but providing some other benefit to the miner. Right. In accomplishing this in the environment has a number of big challenges. One is that these are complicated matrices that have lots of organic matter, most of which is not binding to metals. So you have to have analytical tools that can find the one molecule out of thousands or hundreds of thousands that's bound to a metal and identify what that is. The soluble metals are usually pretty scarce compared to the concentration of organic matter in these systems. And then you need to have analytical approaches that are very clean and are not contaminating not contaminating samples with metal that's coming from the analytical system rather than the environment or sample that you're studying. Right. So there's a lot of evidence that these chelation processes are very tightly controlled in environmental systems. And one piece of evidence for this is just the sheer number of metallophores that have been discovered from microbial cultures. So I love this slide because it gives you a really great example of the of convergent and divergent evolution that's driving strategies for mining, in this case, iron from the environment. So all of these structures here are known microbial siderophores. They're molecules that are produced when certain bacteria are iron deficient, and then those molecules will dissolve iron from the surrounding environment and then facilitate their uptake. And the uptake of these specific molecules requires specific transporters that recognize that molecule. So you can see examples of molecules that look completely differently. These are like desferoxamine E here and petrobactin. The functional groups that are binding to the iron are completely different, right? And these are essentially pathways that evolve completely independently to solve the same problem. Right. And then you also have examples of divergent evolution, right? Desferoxamine E, G, and B, where there's slight structural modifications to that compound, that arise from the evolution of those biosynthetic pathways, right? So this diversity of different molecules nominally doing the same function is pretty clear evidence that the environment is selecting for organisms that can do better and better and better for mining, dissolving the elements that they need selectively enough to facilitate growth. So the organism that's most efficiently able to get the metals that they need is going to grow as a selective pressure. So we can see from some really elegant experiments looking at metal dissolution that these metallophores, even if they're perhaps thought to be used to mine a specific metal, often are not very selective for the metals that they can actually bind in the environment. So Walter Schenkfeld has done a really amazing set of experiments like this one, where he's taken some of these microbial or plant metallophores and added them to soils and actually looked at the kinetics and the binding properties of the metals that they are ending up dissolving in solution. So this is an example from a paper where they took soil from an alkaline environment. This is a clay-rich, carbonate-rich soil that has very low organic matter. And when they add deoxymugenic acid to it, this is a grass phytosiderophore that's produced by many grass species to facilitate iron dissolution and uptake. Right, we can see that over time, the addition of deoxymugenic acid is increasing iron dissolution. So in that first panel, we're looking at concentration of iron dissolved over time. If only a reductant is added, a ascorbate, nothing dissolves from these soils, right? If the siderophore is added, over time you see that the iron is being dissolved. And then if a reductant is added as well, then you see a faster release of that iron. So there are these synergistic effects between redox processes and chelation processes, right? So in addition to iron, though, you can see that large concentrations of other elements are being mobilized by the same molecule. So over time, the amount of copper increases, the amount of nickel increases, cobalt increases, manganese increases very quickly, especially when deoxymugenic acid is added and then drops off. And then zinc exhibits something similar where there's some intermediate characteristic where zinc is going up and then down due to the addition of this deoxymugenic acid. So one of the kind of interesting take-homes of experiments like this is that the metals that are leaching from solution are varying over time and then also influences by the addition of reductants. So there's some important characteristics of selectivity, right, in dissolving the metals that the organism needs to grow. So how can we take this knowledge that there are lots of organisms that can produce lots of different chelators found in the same environments, and many of those chelators can potentially bind to different metals, and start to understand in the actual environment which chelators are important for dissolving specific metals. And so we started to look at this in the context of alkaline grasslands, asking the question, how do native species mine metals from their environment? So we got soils from a series of grasslands that had been undisturbed for long periods of time. So the grasses, crops that are growing in this grassland were native, adapted to those soil conditions, as were the microbes that were present there. These are interesting environments because they're representative of about a quarter of all farmable lands across the world. And under these conditions, where pH is high, things like iron and zinc are hard to solubilize without some sort of chelation strategy. So crop deficiency of these metals ends up being a big problem. So there's a lot to learn about how native organisms are handling the uptake and dissolution of these elements. So we use mass spectrometry-based methods to identify and quantify different metal forms in these environments, with the goal of figuring out which organisms are producing specific molecules that are binding to specific metals. And so the way that these analyses work, so we have a liquid chromatography system that has a trace metal clean platform. So it has no stainless steel wetted parts. That gives us a very, very low metal background. And we know that when we're analyzing samples, we're not introducing additional metal just through our analysis. We use this to separate the different organic molecules that are present in pore waters from these soils. And then we can detect metals that are bound to the separated molecules with inductively coupled plasma mass spectrometry. And so this is an approach where the molecules are eluting into the instrument, they're burnt up and atomized and ionized in a hot plasma, and then those metal ions are detected by mass spectrometry. So this is essentially acting as a sophisticated metal detector. And so when we take one of these alkaline soil samples and separate different molecules over time, in this case, we're looking at how much copper is bound to elements eluting over time, and we can see chromatographic peaks that are eluting that correspond to the individual molecules that are bound to copper. And by integrating the peak areas and calibrating that against metal standards, we can quantify exactly how much metal is bound to the different molecules. Right? And at the same time, we can get metal information about many different elements at once. And then to identify what those complexes are, we can take the same chromatography and couple it with ultra-high resolution mass spectrometry, either Orbitrap mass spectrometry, or if you're fortunate enough to work with EMSL, you can do this with 21 Tesla FTIC-RMS, which is quite an amazing tool for this. And so what this allows you to do is detect the mass of the intact molecule with very high mass accuracy, and you can resolve isotopic fine structure. So that allows you to confidently tell at any of these retention times, we know there's a copper complex eluting where we have a peak, right? There must be a corresponding organic copper complex. And these are very complex samples. There are thousands of things eluting at any time. But if you have enough mass accuracy and can resolve isotopic fine structure, you can identify based on mass defects and detection of isotopologues the mass of the metals containing complex. So today we use software developed by EMSL, CoreMS, to help us annotate molecular formula to the organic metal complexes that we detect. And then we can get fragmentation spectra for those organic molecules and then identify what they are. So in the case of these grassland samples, right, all of the major copper complexes corresponded to different flavors of plant phytosiderophores. We have deoxymugenic acid in this black curve. We have mugenic acid, which is a similar compound with hydroxyl group that can be in two different positions, giving rise to two distinct isomers that we can detect chromatographically resolved. And then we have nicotianamine. This is a structurally related molecule to mugenic acid, where a hydroxyl group is replaced with an amine next to the carboxylic acid, the final carboxylic acid of the molecule. And then finally, we detected this novel set of molecules that seems to be a hydroxylated version of nicotianamine. Right? So it's nicotianamine plus a hydroxyl group, and that can be at two different positions along the carbon backbone of the molecule. Okay, so we can then start to quantify how much metal is bound to each of these different molecules. And I mentioned that we can do this for many elements at the same time. So here's a trace of some of the other elements in the same sample, iron, nickel, and zinc. And again, we can identify the specific molecules that correspond to each of the distinct peaks. So those nicotianamines and mugenic acids account for a lot of the peaks in this sample. And there's also a major iron peak that corresponds to a fungal compound, ferrochrosine, which is a fungal siderophore. And so we can kind of take all the data from the previous slide and digest it into something like this that shows the absolute concentration of metals in the pore waters of these soils that are bound to different molecules. And so we can see that there's a range of selectivities of these different molecules. If we look at what's bound to ferrochrosine, really it's just iron, right? And we can see that of all the species present in the sample, ferrochrosine is binding to the most iron. Then we have deoxymugenic acid and mugenic acid. These are these plant phytosiderophores that are thought to be important for mining iron. And we see that they do bind to iron in these soils, but they're also binding to other elements, nickel and copper as well, right? So they're not as selective as the fungal siderophore pathway. And then we have other molecules like nicotianamine and hydroxynicotianamine that are preferentially binding the softer elements, nickel, copper, and zinc. So small structural modifications can make big differences in terms of what metals these molecules are binding to. One of the surprises in this study was that we were able to see a lot of plant compounds, we were able to see a lot of fungal-derived compounds, but none of the metals seem to be bound by bacterial compounds, even though we know that lots of soil bacteria can produce metal-binding molecules. And so we were curious about what sort of molecules might bacteria from these soils be able to produce, right? And it turns out that they're very willing to give up their secrets. So if we took a small amount of the same soil and we made an enrichment where we added a carbon source, in this case glucose, and then we added nitrogen and phosphorus, but we forced the bacteria to dissolve the metals present in the soil as their only metal source, they end up producing a huge suite of different metallophores that we can effectively detect and identify with mass spectrometry. So we took this enrichment sample and used liquid chromatography coupled to 21T FDIC-RMS at EMSL to get accurate masses and fragmentation spectra for the molecules that we were able to detect. Almost all the molecules that we detected ended up being siderophores in this case, or metallophores in this case, that we found bound to iron. You can see actually four families of molecules. There's the arthropactin family, pyroverdin family, enterobactin family, and furoxamine family, all coexisting in the same soils where we were detecting those phytosiderophores and fungal siderophores, right? So within soils, there's a tremendous diversity of potential molecules that can be used to solubilize metals, but only certain molecules seem to be expressed at any given time. Right. And so we can start to see, this molecular view of metal dissolution in these grassland soils and find that particular phytosiderophores are responsible for solubilizing some of these micronutrients like nickel, copper, and zinc. Others are capable of binding to some of the harder metals, and then fungal siderophores are quite selective. And so one of the things that I think is really interesting about this is that we have a pretty good idea of what organisms are producing these molecules, but we don't have a great sense of what organisms are consuming these molecules. As I mentioned, we were surprised that the bacteria present were not producing their own siderophores in the same system. And so we did some follow-up studies on this, looking at how Pseudomonas bacteria, these are plant growth promoting bacteria, interact with grass species that can produce these phytosiderophores under iron limited conditions. And we found actually that when you grew them together, the bacteria didn't produce its own bacterial siderophore, these pyroverdines, but it instead consumed phytosiderophores that were made by the host, right? And so there are probably some very important cross-kingdom interactions related to how these molecules are taken up. And many of the pathways are still unknown. Okay, so in many cases, we can identify the specific metallophores that are present in the environment and are important, but there's a huge dark metallome that we've become really interested in. And these are molecules where we can detect really clearly metal complexes. We can get accurate molecular formula for them, partial structures based on fragmentation, but they just don't match the mass or formula of any known metallophore, right? So they're orphan molecules that we're not sure why they're being produced in the environment. So this is just an example of lots of different types of organic molecules that we can detect as sharp chromatographic peak for different elements in different types of samples. These are all actually marine samples where a lot of our work was conducted. And so as one example of that, here's an orphan molecule that we detected along the coast of Oregon that was bound to copper. A very large amount of the copper in this coastal ecosystem was bound to these particular molecules. We got beautiful fragmentation data for these molecules. It seems to have heterocyclic nitrogen functional groups that are binding to the copper. We don't have a full structure of the molecule because, again, it's an orphan molecule that hasn't been detected in cultures before, right? So one of the things that we've started to think about in these cases of orphan molecules is if we can start to use environmental context to understand their relevance. And so we can map out the distribution of these molecules across the coastal ecosystem. And what we find is that they're particularly prevalent in waters that are right near river samples, river entry points to the ocean. So this is showing 3 different stations that are closest to the mouths of rivers along the Oregon coast. And they're also hotspots of these particular metallophores. And then if we look at the concentration of these metallophores, these copper binding molecules with depth, we find that they're really associated with the top layer of the water column where we have the most intense chlorophyll. This is where algae are growing and binding to that copper. So when we sample in the river itself, we don't find any of these molecules. So these are clearly being produced by those surface ecosystems growing near the mouths of rivers that are complexing the copper. And it's still a question about whether these are actually being used to facilitate the uptake of that copper or perhaps even to detoxify the copper. So copper is one of these funny elements where you need enough of it to grow, but too much can interfere with other pathways. All the copper in the ocean and most environments is complex to organic matter. And so clearly the uptake of this copper is being carefully regulated, right, to make sure that enough is being taken up, but not too much. In other cases, we found metallophores for things like copper that are less selective. So this is an example of another copper complex that we discovered in the middle of the open Pacific Ocean, the oligotrophic gyre. And in this case, this molecule was bound not only to copper, but also to nickel, right, which is different than the complex that we described before. This also seems to be a compound that has a lot of heterocyclic nitrogen. So that seems to be a recurring theme of these molecules that are prevalent in the environment, binding to more of these divalent metals. Okay, so at the end of the day, to really learn what these orphan molecules are doing in the environment, you need to get the organism in culture and understand the conditions under which the organism is producing those molecules and utilizing them. And so we've started to do that by basically high throughput screening of metallophores from isolates in some of these environments where we detect interesting metal complexes, right? So we can plate out these types of enrichment cultures or natural samples to grow different bacteria. And then with mass spec, we can pretty quickly screen hundreds of colonies and use some data mining tools to find metallophores and novel ones for different metals, right? And so one of the most interesting cases has been this zincophore that we detected in some soil samples. This is a molecule that we found bound to zinc in a system where many other metallophores were binding to copper, to iron. We were able to actually isolate the producer of this molecule, and it ended up being a strain of Pseudomonas, a common soil bacterial strain. And then we were interested in understanding why this molecule was being produced. And so we grew the organism under a range of different zinc conditions and tracked when the molecule was produced. And it turns out that it only makes the molecule when you starve it for zinc. So this is showing a set of growth curves with different zinc concentrations. It's really hard to do these experiments and actually limit things for zinc because there's a lot of zinc contamination in the lab. So even when we don't add any zinc, there's a little bit of residual zinc that gives us a little bit of growth. But you can see that as we add more zinc, we're getting better growth. And then once we get 500 nanomolar or 5 micromolar zinc, we have replete conditions, right? Maximum growth rates. And then the molecule itself, right? When we look at those high concentrations, it doesn't make the molecule. And then at these lower concentrations, when it's zinc starved, it produced the molecule. So my postdoc, Karl Romanowitz, did some transcriptomics on these samples under zinc replete and the zinc deplete conditions to find what genes were differentially regulated. And so it turns out when you starve these organisms for zinc, they upregulate basically two biosynthetic pathways. One is for zinc transporters, just trying to get more inorganic zinc if they can. And the other was a particular biosynthetic cluster. So it turns out that this is the biosynthetic cluster, right, that is producing the zincophore. And so Carl was able to go through the gene cluster and basically determine how the molecule is biosynthesized. So there are genes here containing a non-ribosomal peptide synthase that basically stitches together some amino acids and then tailors them into iron-binding functional groups. So it connects a threonine to a cysteine, cyclizes both of those, connects it to another cysteine, and then repeats that step. So we end up with 2, and then a final cysteine is added by this last gene, giving us this set of ring structures here, right? And then this structure is also consistent with some NMR data that we were able to collect on the same molecule. Right, so looking at the biosynthesis pathway for this molecule, it turns out that that first gene, wardenbactin E, we're calling this molecule wardenbactin, is almost identical to the pathway to the gene that makes this molecule on the left here, pyocheilin, which is a siderophore that's used to mine iron in Pseudomonas aeruginosa, right? And so it turns out in this biosynthesis pathway for the zincophore, the gene regulation was swapped. So instead of being regulated by Fur, as iron regulator, it's regulated by ZR, a zinc regulator, and then a few extra genes are added to tailor the chemistry of that molecule. And so presumably, right, this molecule is more selective for zinc over iron, and that's an ongoing question. So in general, again, the selectivity of these metallophores is something that we don't have a good sense of just based on the structure of the molecule, right? And I think there's a lot of need for better measurements of metal binding selectivity and high throughput measurements of metal binding selectivity of these types of molecules to understand, which metals are they really capable of going after in the environment. Wouldn't it be great if we could just take a structure and then, from that predict accurately what metals it will bind under different conditions? That would be very useful for developing selective chelators for critical mineral mining or separation, for example. So it turns out when you are able to identify a chelator biosynthesis pathway in the environment, you can then start looking for it elsewhere. And it turns out that the biosynthesis pathway for this Wardenbach and zincophore is actually quite widespread. The entire biosynthesis pathway is present in other isolates from the soils, also isolates from the open ocean. Nitrococcus mobilis is a marine nitrifying bacteria, and also from blood isolated organisms, right? And so the need to mine zinc from the environment is quite prevalent, and I think there are probably a lot more of these molecules that are selective for binding zinc. It's just that we have to discover what they are and who can make them. Okay, so to summarize the talk, we have ecosystems that produce a really wide range of metallophores that seem to be governing the dissolution uptake of selective metals from the minerals in the environment, right? So it's really important that organisms dissolve enough metals, but make sure that the right metal gets into the right enzyme and in the right amount. The strategies that are done to liberate metals from soils seem to often involve cross-kingdom interactions between plants, bacteria, and fungi that can produce and take up these metallophores. So, organism is mining metals, but really that metallophore becomes a public good that can be taken up by anything that has the right biosynthesis pathway. And so I think there are a lot of really important interactions that are governed by, keystone microbes that liberate the metals that an ecosystem needs and then, feed that to the broader community. We find a lot of evidence for that also in the ocean. And then finally, there's a large diversity of these abundant orphan molecules in the environment that are binding to metals, and they have no known roles or producers right now, but they're probably doing exactly that, regulating metal dissolution and uptake in really important ways. So going forward, I thought there are a couple of really important existing knowledge gaps that are relevant to the critical mineral thrusts that EMSL is putting together. So one basic question is, what are the role of metallophores for selective uptake of elements beyond iron? And so I think there's an opportunity to identify more of these metallophores and the taxa that they produce them from a wider range of soil environments, right? Particularly from materials that are enriched in critical minerals, right? So one of the rate limiting steps of this is, the screening I think can be fairly fast, but then actually getting to full structural elucidation of some of these molecules is challenging, right? Because it requires the coupling of multiple analytical tools. Another question is, what is the elemental selectivity of metallophores under different conditions? And I think there's again, a really great chance to develop high throughput methods to screen for metal binding molecules that are selective for certain metals over others under specific conditions. And this requires the development of really careful analytical approaches to be able to develop comparable data. But I think that's possible, and it's something that we've been working at in the past few months in our lab and making good progress on. Right. So I think if we can do this in a high throughput way, it has the potential to start to develop the structure to selectivity knowledge that we need to start designing selective chelators that could be used, for example, in industrial scale metal separations. So, one of the challenges in most metal separations is the efficiency of these chelators that are used, for example, in liquid extractions. And part of the challenge of developing better ones is that we're not very good at predicting selectivity for changing organic molecule structure. And then finally, there's a really big knowledge gap related to what genes and species are actually responsible for the uptake of metallophores, right? So when an organism is releasing a metallophore into the environment, there's the potential for other organisms to be able to take that up. And I think if we can understand that, it gives us some targets for enhancing metal accumulation in biosystems. For example, nickel hyperaccumulating plants, they don't solubilize the nickel themselves, right? But maybe certain nickel solubilizing pathways uh, can allocate more of that nickel to a hyperaccumulator, right? So if you can grow something in the same environment as those hyperaccumulators, you could increase accumulation. But to do that rationally, you need to be able to understand the pathways. And with that, I'd like to thank my research group, especially Peter Bright, and then Nicole and Maria Cristina Alvarez, who worked on the Oregon Coast project. Kate Kuba and Carl Romanowitz have worked on the project related to the Zingaphore producer. And then a lot of collaborators. Much of this work was done in collaboration with people at PNNL, Lily Pasatolik, Janet Jansen, Kirsten Hofbuckle, and Jared Shaw, as well as Sarah Fansler. And with that, I'd love to take questions. Thanks for your attention.
Well, thank you so much, Rene. That was an awesome talk. I think you gave a really neat overview of some of the chemical and analytical challenges, as well as some of the really fascinating biology. First up, we had a few questions in the same sort of theme, so I'll group them together. One of the questions is what structural features are in these siderophores are contributing to selectivity? And at the same time, another question, is anyone using AI to predict synthetic siderophores or to predict properties of natural siderophores, maybe unique or unexpected binding capabilities?
Yeah, those are both great questions. So to the first question, the general trend for selectivity probably relates to the hardness of the ligands that are binding to the metal. And this is the kind of rule that we typically use to think about which ligands are going to be more selective for different metals. So something like a carboxylic acid is a harder ligand than something like heterocyclic nitrogen or like a sulfur-containing molecule ligand, right? So those will tend to bind harder metals to softer metals. But then, I think that it's probably a lot more nuanced than that. And the bottom line is that we just don't have good, careful, comparable data. So to measure metal binding selectivity traditionally requires difficult experiments that are either done by competitive ligand exchange or by titrations that require a lot of the metal ligand, and then they're involved measurements. So oftentimes this binding of one molecule to one metal is done in like maybe one lab in one context, and selectivity might be determined by comparing measurements made in different labs at different times under slightly different conditions. And so oftentimes those numbers aren't even necessarily super comparable. So that relates to the AI question. I think in general, we don't have a great data set for metal binding selectivity, right? We know a lot about how synthetic chelators are binding to metals and about their selectivity, but that's a pretty narrow chemical space. And what you find is that evolution is telling us that there are much more elaborate chemistries that you can develop probably to get better selectivity under different conditions. And we just have to first get some preliminary data on selectivity. But then I think using computational tools to model that, right, and then get to the point where we can model it and then predict unknown structure functions would be really powerful for so many applications. One of them being, relevant to critical mineral separations.
Cool. Thank you so much. So another follow-up question is to do with the orphan metallophores, which I think got a lot of interest. Were you able to identify any biosynthetic pathways for these? Was DNA or RNA collected at any of these sites?
Yeah, so in the other examples that I presented, we didn't have genomic data. This is something that we need to do more of, right? Coupling the collection of the metallophore uh, detection and characterization of metallophores with genomes and metagenomes. Um, But I think doing that also just through like screening of cultures is a really effective way to do it too.
Okay, thank you.
Also
So,
have the isolate then.
cool. So we got a couple of questions about the Warden-Bactin. One quick question is the name from the Warden Soil series?
Yes, it is. So the isolate was from Warden soils. That's right.
Nice. And then in another
Which is
question,
near Pinot. Yeah.
have you looked at the selectivity for the Warden-Bactin or for any of the Metallophores? The selectivity at the level of transport and uptake?
We haven't. So what we're currently trying to do is develop mass spec based methods for measuring just chemical selectivity of metal complex versus other metals. So we've wanted since finding this molecule to measure its selectivity for iron versus zinc, but it's hard to get enough of the material to do the classic titrations. And so it's really motivated us to start looking at other methods. So using mass spec to measure the ratio of metals bound to a molecule under different conditions seems to be a pretty effective approach. So I think we'll hopefully have updates on that soon.
Nice. There was a couple of questions maybe to do with more unusual types of metallophore producers. So one person was asking, have you looked at metallophore producers from hot springs? And another question, several of these transition metals are involved in C1 metabolism. Have you any work looking for metals scavenging molecules produced by methanogens or methanotropes?
Oh, these are all great questions. We haven't looked at hot springs. It would be a really interesting environment to look at. In general, when we go to a new environment, we find interesting things, interesting metallophores. Oftentimes, for metals other than iron, they end up being these orphan molecules that are a little tricky to track down, why they're being produced. What was the last question?
So there was the question about the methanogens and methanotropes.
Yeah, yeah. So we haven't specifically looked at methanogens or methanotropes. Interestingly, we sometimes see copper complexes in anaerobic environments, which could be related to these types of metallophores. So bacterial methanotrophs are some of the only organisms that are known to produce a copper binding metallophore to their methanobactins. So that they're probably actively involved in dissolution of some of these metals as well.
Okay, I think we have time for maybe one more question before we have a break. So you mentioned that Pseudomonas may utilize phytosiderophores when grown with a plant host, but those pathways remain mostly uncharacterized. Have you started looking at what those pathways are, or do you have any more information on this metabolite sharing?
Yeah, so we did actually do transcriptomics of the bacteria in those experiments, and there were a handful of genes that were upregulated that were like related to small molecule transport and uptake. So we do actually have a couple of candidates, but we haven't had the chance to follow up on identifying those. Hopefully the
Okay.
project's funded next time. Yeah.
And then just one very last quick question, and I think this is a good one to wrap up on. Is there a siderophore database? And if not, should there be one created?
Yeah, there are a handful of efforts, Herculean efforts that have pulled some of these siderophore into databases. It looks like someone has found one of the more recent ones and posted it as well. I would say that again, we have these siderophore databases, but it's not necessarily the case that all of these are necessarily selective for iron, or even in all cases being used to mine iron from the environment. And then there are probably a lot of other molecules that are more selective for other metals and don't get picked up through siderophore screens. But the siderophore databases are a really powerful starting point for identifying any metallophores. And we use these databases as our way to start trying to identify things that we find in the environment once we have a mass and molecular formula.
So I'm Amir Ahkami, staff scientist with EMSL, currently leading EMSL's integrated research platform for rhizosphere function, as well as the EMSL rhizosphere critical mineral campaign. The short name is Rhizocritical. So later today and tomorrow, I will also be facilitating the breakout sessions for this campaign. It's my honor to introduce our final keynote speaker for the plenary session, Dr. Scott Angle. Dr. Angle currently serves as the Senior Vice President for Agricultural and Natural Resources at the University of Florida. He holds a bachelor degree in agronomy and a master degree in soil science from the University of Maryland, along with a PhD from the University of Missouri with a focus on soil microbiology. Dr. Angle is a fellow of the American Association for the Advancement of Science and a member of the Academy of Science, Engineering, and Medicine of Florida. Previously, he was a professor of soil science at the University of Maryland, where he also served as the director of the Maryland Agricultural Experiment Station. Dr. Angle is widely recognized for his groundbreaking research on phytoremediation and phytomining. So please join me in giving a warm welcome to our keynote speaker, Dr. Scott Angle. Scott, stage is yours.
Well, thank you, and I appreciate the opportunity to be here with all of you. And as the others have said, I'm sorry it's not in person, but maybe at some point. So thank you for your welcome. I want to walk you through phytomining and phytoremediation, how this technology developed, because that is going to reveal a number of gaps that I think there's still opportunity for improvement with this technology. Again, I'm going to talk about the past, present, and future, but I'm really going to try to focus on the gaps. This all began in my career and several others who were instrumental in developing these technologies using plants to extract metals from soil. But I want to tell you this quick story. This was in the 1980s where Rufus Cheney, who is considered the father of phytoremediation and phytomining, was called up to a small town in Palmerton, Pennsylvania, where they were unable to raise ponies. These ponies would, within about 3 months, die. They were unable to stand properly, and since they weren't able to nurse, they would eventually just starve to death. And they had for years a veterinarian in that town who didn't seem to think that this was a problem, but he retired eventually. They got a new veterinarian. And the first thing he said was, there is something seriously wrong in this town. We need to figure out what it is. We all assumed that it was the zinc smelters called the Horsehead Mining Company, again in Palmerton. This is a really old picture, but in the upper right-hand corner, you can see the smelter itself. But everything from the smelter and downwind was dead. And it was due to a variety of different contaminants, primarily zinc. So Rufus brought me into this project. That was my beginning. And we eventually determined that it was a zinc-induced copper deficiency, and copper is needed to form cartilage between bones. And these poor animals were in so much pain that they simply couldn't stand anymore to nurse. So that led us into thinking about how could we remediate these areas. It was large areas, sometimes dozens of miles downwind, that were impacted. And so we started to think about what might be needed. And that's when the whole idea of phytoremediation, phytomining was discovered. And I'm going to give a lot more people credit here for this, but I think all of you know that this is a use of plants to extract metals out of soil, including some rare earth elements, and that those metals can either then be landfilled if they are low cost, but if they have a higher enough value associated with them, like nickel that most people are working on today, you can actually sell the nickel and use this as a company to make products. The remediation, it's an expensive process. It's about $3 million an acre using a traditional dig and haul and landfill process. Okay, so at the same time, as we were developing these needed technologies to clean up polluted soil, there was an individual, his name was Robert Brooks. He was from Massey University in New Zealand, and he was discovering these plants called hyperaccumulators that had this very unusual ability to take up significant quantities of metals. Robert had passed away a number of years ago. This was probably back in the 1980s, and that's it's a plant called Berkea, which was one of the early nickel accumulators. So this technology started to blossom then with the discovery of these plants. And these are odd plants. We don't quite know why they accumulate heavy metals. Sometimes there are a number of people that think it protects them from disease, certainly insects, grazing by deer, weed competition. It can pump up metals out of the soil and shed the leaves, and it creates a toxic zone around the base of the plant. But no one knows exactly for sure why these plants are doing what they are doing. But it's certainly interesting. So this is a picture of Rufus Cheney, and this was really the first work on phytoremediation now, not phytomining. This was a plant called Thalassemia caryolescens. It was from southern France, and it was initially identified as a zinc accumulator, and it could accumulate 2 to 3% zinc in the leaves. But as you can also see, it's not a particularly tall plant, and for phytoremediation, phytomining to be effective, you have to be able to harvest it just like you would a normal agricultural crop. And it became somewhat problematic, although there were, and the other problem was that the value of zinc was low. And so no one really cared that much about it. Turns out another variety of this plant can extract cadmium from soil, same genus and species, and it was explored briefly for cadmium phytoremediation of soils. There are millions of acres in Japan, for example, that are contaminated with cadmium. These are typically rice growing areas. So we thought about using that, but the scale of the problem and the time frame needed to clean up these soils just wasn't practical. Once you get the legal process involved with the Superfund site or other contaminated site, you don't have a long time. You can't wait 10, sometimes 15 years that phytoremediation is needed to clean up the soil. They want it done that summer or that year. And so going back to that dig and haul mechanism turned out to be really the only way that was practical. And so it created a problem for a number of years as a result. These are some of the early innovators. I'm going to come back to this of phytomining, phytoremediation, but here's Rufus. He was a plant physiologist on this team. Here's me in the back. I was kind of the agronomist. I'm the one who understood how to grow these plants. This is Alan Baker, one of the people who discovered many of these. He was from Sheffield University and then University of Melbourne later. Roger Reeves from Massey University. So that was kind of the small group, and these are other people who had joined on. This was from a meeting back in Siena in 2006. So it's not a new technology. Here's another close-up picture of Thalassemia caryolescens, zinc accumulator, but also removes some cadmium. But as I said, the value of zinc was not very low. Certainly cadmium was not widespread enough to justify full development for phytoremediation. So the technology kind of stalled for a while. There was probably a decade in maybe the 2000s where there were some projects, few on nickel, a couple on arsenic. Cadmium was still in the public domain as a concern, but there really wasn't much going on right now. There was another problem that occurred that I want to explain next, and this was a very unfortunate thing that happened. We all realized around the year 2000 that zinc was not the metal if you're going to try to develop a mining technology. It had to be something with greater value, and the obvious one was nickel because there were lots of nickel hyperaccumulators that were found. So it's a valuable metal. There are probably millions of acres around the world with high nickel soil. These are called serpentine soils. There are plenty of them in the United States and Southern Oregon and Northern California. As I said, most of the hyperaccumulators found to date were nickel accumulators. So it seemed like a great opportunity to develop a new industry. So our group of Alan Baker, myself, Rufus Cheney, Yin Lee was a postdoc who's standing in the middle of that field there. This was a field of the nickel accumulator in Southern Oregon. It's called Cave Junction, and it was growing really well until some of these plants began to escape. They were well adapted to this Mediterranean, kind of cool Mediterranean climate. They escaped from the research site It's called, this species is called alyssum, or it was called alyssum back then. It escaped and became a weed. And that really then put the spike in the heart of this entire industry. Because to think about this, this was a invasive plant. It was a weed. It was also poisonous, and it was from another country. And so it created a lot of concern in that region. And as I said, it then it become very difficult to do this research in the United States. Now, research continued in other parts of the world, particularly in Albania. There's some areas in Indonesia, New Caledonia, where the work continued, but not in the United States. Next, what I want to talk about is, I'm going to get us over this hurdle that I just talked about invasive weeds from another country. But I want to start working my way into how do you take a plant from an area around the world and turn it into a new agricultural crop. Just think about corn or soybeans or wheat and all the work that went into, sometimes hundreds of years, in the case of wheat, thousands of years of breeding and improvement. How do you do this within a couple of years? As I said, you have to think about the weediness of the plant, the fact that these plants are nickel toxic. And there's a lot of people working on genetic modification. So as I think about this from a regulator's perspective, so you bring them a weed from another country that's invasive, that's genetically modified, and it's poisonous. Their heads are going to explode when you try to get that moved into another country. Now, most of the hyperaccumulators, though, have been found in other countries than the United States. And I'll give you one good example. For the same species of alyssum, we at one time found a variety of alyssum that could accumulate about 4.5% nickel on a dry weight basis. That's been the highest nickel content ever discovered for a plant. So the way you find these plants, at least in the past, you'd go to an area where you think these plants will exist. You have some knowledge of botany and plant species and good idea which ones you think might be hyperaccumulators. You collect the leaves, you bring them back to the laboratory, you test them for whatever metal you're looking for. And if it comes up that it's high in nickel, then you go back to that site the next year and hope that it's in seed, or hope that you can dig up the plant and bring it home with you. Well, you can't bring seed and you can't dig up plants anymore because of new treaties globally to restrict plant movement. That's almost impossible. But the sad part of that first part of that story I was just telling you is when we went back to that site, it was an island in the Aegean Sea. There was now a shopping mall on top of that where that plant used to be, and we never found it again. So it was at that time very complicated, and it made it difficult for this process. But as I said, our goal is to make this as commercially viable as possible using existing equipment, nothing special. And so we treat this plant just like we would alfalfa or hay. You sow it, you broadcast it, you grow it, you probably apply fertilizers, maybe a little bit of weed control. But once it's done growing, you harvest it here with the sickle bar. You can then bale it up into hay just like this. So those are bales of alyssum, very high nickel content. The next step then is to extract the nickel back out of that bale of hay. I've been fascinated by the two earlier talks as there's been a lot of information shared about how you can think about extracting nickel out of this. This is clearly 90% organic matter in these bales of hay. So the first thing we would typically do would be to burn the hay. And early on, we just took these bales of hay and we shucked them into a smelter. There was still an existing smelter, an Inco, International Nickel Company, not too far from where we were phytomining. So we chucked those bales into the smelter itself. And out the other end came relatively pure nickel. It wasn't pure enough to be used yet. But as we worked with International Nickel Company, we realized that was not going to be an effective method for extracting nickel from biomass because it allowed some contaminants to remain in the nickel, but it also fouled up the entire smelting process. And so these big companies weren't willing to sacrifice these hugely expensive smelters for a relatively small amount of biomass to be input into it. And so that raised for us a very significant question. We've already heard some ideas about how this may happen. But the problem was that there are contaminants in the plant material, a lot of calcium, sodium, potassium were difficult to extract, to separate the nickel from. So there were also air pollution concerns with the smelting, or excuse me, the incineration process itself. So there's a lot more work needed on if phytomining is ever going to move forward. We need to have new methods for extracting these metals out of the biomass. We're lucky in the United States that we have very large acres. In fact, we have millions of acres in the U.S. alone. These are the red areas, both on the East Coast and the West Coast. They're always on the western side, and this is found regardless of where you are around the world. These areas of high nickel content, in this case, are always found on the western side of a mountain range. So in California, it's the Sierra Nevadas. East Coast, it's the Appalachians. These are ultramafic rock outcroppings. The rock minerals are, well, they're primarily olivine. So because of all of this, and because the technology had kind of faded away for maybe 10 years or 15 years, not a whole lot was done. But more recently, as a result of the inability to bring in plants from other countries, but also simply the higher value of nickel, we decided to reinvestigate a plant that we had discovered several decades ago called Streptanthus. It's a very small, kind of pitiful looking plant. Has pretty flowers, but it grows on the rock outcroppings of the Sierra Nevada foothills. But when we had found this initially, we ignored it because it wasn't big enough. You need the phytomining of any material is based upon the content of that mineral, of that element in the plant, times the overall weight of the plant. And we had good nickel content of these plants. We simply didn't have enough biomass, so we ignored it. But anyway, fast forward 15, 20 years, we decided to reinvestigate this plant. We weren't very hopeful that it would have any practical value, but because of the way things had changed and because of as rare earths and other elements became more important, we decided to give it one more look and maybe another try. But here's one of the things that was never realized as people were exploring for these plants. Most of the explorers for hyperaccumulator plants have made a huge mistake. They're looking for plants that grow in high metal soils, and I'll show you some pictures of them. But those soils can support a lot of other plant species, and there simply isn't the evolutionary pressure on those soils to produce the characteristic of hyperaccumulation. The only place that hyperaccumulators have ever been found are on these rock outcroppings that you see here. This one's in Southern Oregon, one is in California. As you can see, there's not much growing on them. Many of the plants here these are not hyperaccumulators. They're brush that have adapted or adopted some other mechanism for survival. But once in a while, and unfortunately you can't see it on this picture, you'll find one of these very pitiful looking, looks like just a small stick, about a foot tall, sticking up out of the rock. That would be a true nickel hyperaccumulator. And so those are the plants that we looked for. So again, just to reemphasize that, plants are only found on mineral rock outcroppings. So if any of you are interested in searching for hyperaccumulators, these are the types of areas that you do it in, not more traditional open ground, true soil environments, because again, the evolutionary pressure for the development of hyperaccumulator simply did not exist there. The other thing is that if you take a hyperaccumulator that came from those rocky areas and try to move it to a true soil, even if it is a high metal soil, it can't survive. It doesn't have the same type of mechanisms that allow it to compete with other plants. So it gets overgrown by grasses and other broadleaves pretty quickly, and simply not adapted to anywhere but those rock outcroppings. This is all hard work to do. Many of these areas are very remote. They can sometimes be, I'll show you here in just a minute, some of the hazards of this. But there are, I believe, still a number of hyperaccumulators of a variety of different metals yet to be discovered. So hopefully in the United States, given that we can't bring plants from other countries to the US very easily anymore, but hopefully in the US we still have some more to discover. And so this is my fun picture of this for my presentation, and that this is what graduate students, why we have graduate students, is to tell Jacob to run up that mountainside and find a plant. Well, I feel like that old wildlife show. Well, Jim scampers up the mountain to, corral a lion. I will stay down on the bottom of the hill and have a bag open to put that plant into when you get. So it is hard work. It can be somewhat dangerous. We've had plenty of encounters with wild animals. We have had plenty of encounters with rattlesnakes as well. We never had our camera ready when we encountered a rattlesnake, but a lot of these plants grow in places that rattlesnakes seem to love. And so worker safety, whether it's a snake bite, and we typically wear gaiters on our legs to prevent snake bites, or a fall in a remote area. What are you going to do if you don't have cell service? So we have a lot of protocols that we put in place for making sure that we stay safe during these collection trips. But this is kind of the first step. When you find one of these plants and you now know it has a high nickel content, you need to then think about how do you domesticate it into something that could be commercially, or if it's for phytoremediation, have practical environmental value. And what we often find is that scrawny stick-like plant growing on those very rocky soils, when you get them back into better soil, like you see here, and this would be a serpentine soil that this plant is growing in, then you have multiple stems, grows much higher, a lot more biomass. And for us, this was the eureka moment when we found that something that we really had no hope for ever growing into a practical technology did start to grow much taller. And I'm going to show you a picture in a minute, but it turned out that what grows on those rocky skeletal soils can be very different from what grows in true soil. Now, when we grow it in soil like this is a high nickel soil, you have to control competition. You've got to use herbicides to control grasses in particular, sometimes even broadleaves, and we've got some herbicides for that we've discovered over the last year or so. So this is a big gap. How do you take a weed from a very remote area, sometimes never described before? How much fertilizer does it need? How do you control pests? Metal solubility, I think, as you have already seen, is often controlled by pH. You need to amend the pH of the soil. How do you control weeds? How do you harvest a plant? How do you plant these plants? The seeds are often very, very small. Do you need irrigation? None of these are known when you go to domesticate into a commercial application or commercial technology. And so this is what my laboratory spends most of the time on, is simply figuring out how can we grow these plants into something that has some practical value. So in doing all that, here's now what one of the plants looked like. That was a single stem. Remember that first picture I showed you of a very small stem, almost no leaves, no biomass. This is one plant now that is growing in better soil. This is, we're doing this in the foothills of the Sierra Nevada mountains. If you know that area, it's either Jackson or Murphy's is the area where these plants are now being tested out in the field. So this was the first field demonstration of nickel phytoremediation using this particular plant species. Again, we would have loved to have used alyssum or the yellow species I showed you earlier, but it's just not possible, partly because of some of the mistakes we made early on, but also some other management issues that had nothing to do with us. we had to go with a California native plant. Now, even getting this plant into Oregon may be a problem. So we're addressing that now with the Department of Agriculture in Oregon to see what the limitations are for that. I'm not sure it's even possible to get it across state lines. So going from a wild plant to the greenhouse, which is where we do all our initial testing, to this is the field test where I showed you that larger plant there just a minute ago. It's a beautiful area. These are high nickel soils in this area, high cobalt soils as well. This soil has about 2,800 parts per million nickel in it. The plants that we're getting from these soils we're trying to get up to 20,000 parts per million in the plant, dry weight of the plant. We're still at about 12 to 15,000. So we've got some more, got some greater need for either manipulating the soil or manipulating the plant. We're trying all kinds of things right now to change the dynamics of plant growth, either through genetics or cultural practices. But our goal is to get a little bit higher than we are right now. But we're pretty close to having a plant that can be practically used. And this is only 1/10 of an acre, but this is the first demonstration of phytomining using Streptanthus in the world. This is California with the 2 graduate students I'm working with. So this was a relatively significant project. We harvested these plants in June of this year, and next month we're going to plant about 1 acre of that same species. So we've collected a lot of the seed. We allowed these plants to go to seed, and we're going to try to scale it up tenfold every year for the next couple of years. So what's next? This is risky. Whether or not there are plants that can take up higher than average and hopefully very high amounts of rare earth metals. I'm not particularly confident that any of these plants exist, but as far as I know at this point, hardly anyone has looked. I know there's a company, and I believe it may be out of Arizona. There's a little bit of work going on in North Carolina. But so far, no one has found any of these plants. And frankly, there's not a good appreciation for where you would find these plants. The best way to do this, by the way, if you're looking for new plants that can extract anything, is not to go out to the field. It's to go to a herbarium at a large university and check plants there. You can, instead of doing maybe 50 plants a day, you can look at 1,000 plants a day. I'll show you what's made all of that possible here in just a minute. So I'm not very optimistic that there will be plants that can take up any of these elements, but it's worth looking because the reward is high for our society if we can find these plants. So we might as well try. But what has made this so much easier, as I said earlier, we used to collect a leaf sample, bring it back to the laboratory, run it through an ICP-MS, measure metal concentrations, go back to the field, pray that the plants are still there and there's not a shopping mall on top of the plants anymore. But our XRF handheld device, which is relatively expensive, by the way, has made this so much easier. It's not a research device, but it gets us pretty close. It's probably plus or minus 10 to 20% of what we would find through ICP-MS work. So it's a great screen for field work. And we take this gun out with us to the field or to an herbarium, and we can go through lots and lots of plants very quickly. It does many of the nickels that have values associated with them. And it does at least, I just got this information this morning, does at least 6 rare earth elements as well. So we're going to start screening some of our plants here soon to see whether or not any of it is taken up. As I said, I'm pretty pessimistic that it will be successful, but at least it's worth trying because of the risk-reward proposition. I just want to reemphasize this point because I know there are some people listening right now that would like to spend some time exploring for plants. On the right there, that's where you can find hyperaccumulators. On the left, which is it's a high nickel soil, high cobalt soil, but you will never find a nickel hyperaccumulator or a cobalt accumulator on the left-hand side, though, because of the way that these plants have evolved over time. So let me just end here with, if any of you are interested in exploring for these elements, we'd certainly be happy to work with you, but you need 3 people on every team, in addition to graduate students, to climb up the mountains. You absolutely need a geologist with knowledge of hard rock mineralogy, hard rock mining. Where do these outcroppings occur? It's typically desk work, but they can get you out to the right place. Secondly, you need a botanist with local knowledge of plant species. A lot of these plant species are not well characterized, identified. Sometimes they've never been characterized at all. And so having a local botanist with you who kind of understands what's important in that area. And then it's always good to have a phytomining expert who understands kind of the practical side of why all this is working. If you find a really little tiny plant that doesn't bolt up for harvesting, then it may not be something worth exploring. So those are some of the needs. There are a lot of other needs as well. I was listening to the 2 earlier presentations. I think there is a need to isolate, identify, and re-inoculate back into soil, or maybe even coat seeds with nickel or other metal solubilizing bacteria, or maybe even fungi. Our hope in the long run is to create essentially a package where a seed is coated with a little bit of fertilizer, it's coated with a metal solubilizing bacteria or microbe, I should say, and also maybe even coated with something to adjust the pH of the immediate niche of where the seed will germinate, because you do often need to lower the pH of these soils. So that's kind of phytomining and in a nutshell there. And if any of you have questions, there is my contact information. I'd be happy to talk to you any more about this.
We got a lot of questions. I'm not sure if we get time to answer all, but I just start with some. Is the nickel sequestered in certain parts of the plant biomass that could be separated physically from the other parts of the plant? It's a question by Yushi-Kopp.
Yeah, the nickel is in all parts of the plants. It is higher in the leaves than the stems, but practically you need to harvest the whole plant. You need to have as much biomass as possible. And you can, while you may have 20,000 parts per million in the leaves, you may still have 7,000 or 8,000 parts per million in the stems, because the transport process is a continual process during the growing season. You also have high metal content in the flowers. So, and then practically, it would be very hard to separate the leaves from the stems.
Okay, great. Thank you so much. Another question is that in our experience, we see different concentrations of metal accumulation depending on the part of the plant. The root usually holds a lot more than leaves. In the harvesting of the plant, is the root left on site? Is the question from Cecilia.
Yeah, Cecilia, that's a great question. We at this point we are treating it just like alfalfa, where you try to cut it as low to the ground as possible, leave the roots in. It's possible the plants may grow back from the crown in the second year. No one knows this for sure. There's some information in the literature suggesting that, but we don't know that. We thought about that much more with Thelaspi and the zinc and cadmium accumulator, because as you saw in that picture from Rufus Cheney, there's an awful lot of biomass below ground, almost as much as above ground. And that is where most of the metal is accumulate, even higher concentrations. So it's something to look at. There are technologies that I think would make it practical to pick up the root system as well. We have vegetable harvesters like carrot harvesters, peanut harvesters. I think that might work, but that's just something that we've never looked at.
I'm wondering if the search for hyperaccumulators could be of value to discover new metallophores, presuming that the plants or associated bacteria generate them to facilitate hyperaccumulation that could be used in the manner Rene was talking about, instead of accumulation in plant tissues. Has anybody considered research along that line?
No, it's a great idea. I'm not aware of anyone who has done that before. Yeah, I think the association, a natural association between a plant and a bacteria might be an appropriate way to approach that.
Another question, you showed the map of nickel hyperaccumulators on the west side of mountain ranges. Is there no nickel hyperaccumulation on the west side of the Rocky Mountains? Is this based on range of mountain range? Is this based on age of mountain range?
Yeah, these tend to be from tectonic plate elevation. It's possible to predict where you have had appropriate tectonics for that. I don't know why we haven't found more on the western side of the Rocky Mountains. Good question, but there's been plenty of searching done in that area, and I'm just not familiar with anyone finding plants, at least for nickel.
Okay. I think this is going to be the last question. Kind of two questions because the second was follow up on the first one. Are there particular metallophores in soil that have been associated with hyperaccumulator plants? And the second one, as a follow-up to this question, do hyperaccumulators have characteristic rhizosphere microbiomes with high metallophore production capacity?
Yeah, I did some of that work back in the, well, over 20 years ago. We actually filed a patent on the discovery of bacteria that could solubilize nickel. Those patents are long since expired, and the work was not very sophisticated at the time. But yes, I do believe that should be a part of any technology for using plants to extract metals from soil is by discovering and probably either encouraging the growth of those species or inoculating them into the soil and hoping that they survive.
Okay, fantastic. As I said, there are more questions, but I think we cannot answer those right now, but we'll make sure that we deliver those questions to our speaker and he can provide the answers and we can share with participants who ask the questions. Thank you again, Scott. That was fantastic talk and Q&As. Thank you very much for all the participants who actually really very well engaged with these presentations. I think now it's the time to move to the roundtable discussion. We have questions for all the invited speakers. So I pass it to Will to actually pose the first question.
So this question is for Rene, not to put you on the spot. So as we heard this morning, EMSL is a interdisciplinary capability. It's a user facility with 150 different types of instrumentation. We have different science areas, different campaigns, lots of different analytics and capabilities. And obviously, you've worked with EMSL before over the years. So based on your familiarity with EMSL's capabilities and expertise, which challenges that we've sort of discussed this morning do you think seem particularly well suited to EMSL and collaboration with EMSL?
Yeah, thanks, Will. I would say that one of the things I see EMSL as being really useful for is projects that involve like high throughput screening through multiple analytical lenses, right? So, for example, being able to screen lots of hyperaccumulators, right, to generate a database of plants that are accumulating different types of metals, and then at the same time generate complementary data like rhizosphere microbial community and metallophore composition, right? So to be able to, I think a lot of the challenges require multiple data sets that are both, elemental and biological or biochemical. And a place like EMSL is really well set up to construct those, right? And then this can be a library to start finding, targets for deeper study.
Cool. Thank you so much. I will hand over now to Odetta to ask the next question.
Yeah, the next question is for Helen, and I will encourage all the keynote speakers to keep the camera on because this is kind of a roundtable, so we probably if we have time, we can bounce some of these questions among all of you. So for Helen, so thinking about science challenges and opportunities that we talked today, Which fundamental biogeochemical topic would you highlight as being the most impactful to broadly enable applied research efforts and improve the de-risking of CMM recovery from unconventional sources?
The most impactful. I think if we can, well, a lot of our work that has been done in this space has been about characterizing the kind of materials you have and their transformations. I think the impact comes in making that quantitative. So we're talking about rates of transformations and so to enable models of that so that they can then have broader applicability. So I think it's really translating the information into things that are then can be taken into to models.
And then just going to follow back into the idea of being able to apply it, to use this in applied research. Yeah, this is what in your mind will de-risk these efforts if we can get this type of information.
Yeah, that and also getting an idea of how we can translate that into an inventory scale understanding of the resource potential of anything that's like a waste that we're trying to do waste to resource kind of application is like the reserve of that, like the quantity available and the quality of it is really important.
Okay, awesome, great. Amir?
The next question is for Scott. So what is the most important lesson learned from your experience designing and conducting research in the area of phytoremediation and phytomining? For example, the pitfalls, traps, or any deep challenges?
Yeah, well, this is like prospecting for gold. 99% of everything you do fails. And so having patience, having a willingness to be under somewhat harsh conditions for a long period of time. And then as I ended up with, you need a team of a geologist, a botanist, and a kind of an agronomist, I guess would be the best way to put it. And just be patient. These plants exist, but it's a needle in a haystack, and you have to be willing to put in the time and the effort to find these rare plants that often live in hard to get to places.
Great. Just a quick follow-up on that. If you wanna, because I know you had experience working on this area, but you know, using or exploring the microbes in the soil or the rhizosphere of these plant species. If you wanna consider that area specifically, or so-called microbiome-assisted phytomining, and the roles of microbes to make those, you know, minerals available for plant roots to be uptaken, then specifically on that area, what you think would be the deep challenge or pitfall or the lesson learned? Same question, but kind of mainly focusing on the rhizosphere area.
Well, we had done that, as I said earlier. We had found some, I don't remember the species anymore. I think they may have been Pseudomonas. But we would isolate nickel solubilizing bacteria, grow them up, re-inoculate them back into the soil. We didn't have a particularly large increase in nickel uptake. It was maybe maybe a couple hundred more, maybe 100, 200 more parts per million into the upper portions of the plant as a result. There is a downside. It's a lot of work to have to do that. That's why we're hoping to be able to include it with the pellet around the seed itself. But our study was fairly limited. It was long ago. There are much better technologies now for isolating bacteria. And you've heard about some of them today. So I think it's something that needs to be reinvestigated. And it's very possible that this could be an important part of the phytomining or phytoremediation technology. Let me just say, though, you have to balance this as well. This was long ago. We were working on gold in Australia. And we found ways to solubilize the gold. But what was happening was we were driving the gold chelate down into the groundwater, Some of it went up into the plant, so it did work, but we were spreading more contamination around than we were actually harvesting out of the soil. So it made the problem worse, not better. So you have to be careful when you think about solubilizing anything in soil. Not all it doesn't, you don't take up 100% of it into the plant. What happens to the part that you don't take up? Have you now just created a greater problem associated with pollution?
Violetta, have other questions?
Yeah, I think we have one more question, but I'll open them to all of the attendees, all the speakers here, and just take your, just give your response to that. So the question is, if you had now the chance to write a proposal for DOE in your research area, and you have the opportunity to organize a campaign that is multi-institutional, what will be a science challenge that you could identify for that proposal?
I'll go first because mine's fairly simple. I think there ought to be an effort to look for plants that can take up higher than average concentrations of rare earth elements. As I said, I think it's a risky endeavor. It probably doesn't need to be done multiple times, but someone at least someone needs to confirm that they either don't exist and we need to give up, or if we get lucky and find one, then there's a whole new area of opportunity for research.
I think from the standpoint of from our group, I think taking what we know in terms of biogeochemical processes in this, which has typically been in the standpoint of either like earth system, ecosystem science processes or contaminants, and kind of taking that, because there's a lot we can carry over to this idea of resourcing, like work that we know about trace metals. Well, in this case, the critical minerals are trace, but we can actually take what we know and use that information and figure out how to concentrate things and understand their occurrence and reactivity. I think those are the kinds of themes that I think I would go after.
I'm of course interested in strategies for solubilizing metals, and so I would be interested in a program that attempted to take some of these unusual feedstock minerals with lots of heavy metals and took microbial communities from different soil environments, and then basically determined, like, if you allow those communities to evolve in a condition where they have to get all their metals from those minerals, right, what strategies do they end up using, and basically track the microbial community and the pathways that are solubilizing those metals. And I'd be curious to see, if you have one type of mineral, do all different microbial communities converge on the same strategy, or is there a stochastic nature of this that really depends on the starting microbial community? But I think that would provide a lot of basic insight into what would evolution choose as the best strategy for dissolving a certain type of mineral, that's relevant to things that we're trying to get out of minerals.
Campaigns for in situ applications would be different from bioreactor applications. Andrila thinks that there are strengths and challenges to apply towards each of those. So just kind of putting this in the context what everybody said, and if you want to add more to this statement that Andrila put in the chat.
Scott, your mention about the creating a problem with groundwater, really resonates. I think that is a risk of the in situ part, is making sure that you can contain anything that might be a negative impact. The challenge with the bioreactor, with bioreactors as a counterpoint to that, you can then engineer the process conditions, the processes that you want to happen, you can control it better. But then a challenge with that is that if you're dealing with like these mountain locations where, even our grad students, like, we worry about them climbing up the hills, right? I think that's where these, the bioreactor applications are just not feasible. So it's those kind of trade-offs that you have to think about.
I would just like to personally thank everyone for the wonderful talks and very engaging. You could see from the number of questions that we received how engaging were for the audience. And then Will and Amir, do you want to chime in? Thanking our speakers.
Yeah, I just want to thank speakers and participants. The talk was really, all talks were really fantastic. And as you mentioned, I think the level of engagement is really, really fantastic. Thank you all.
Yeah, and I'll just echo those sentiments. Thanks again to our three excellent speakers. I think you've done a great job setting up these three new exciting campaigns that EMSL is launching. For everyone who is listening in, thank you for your questions. If you want to learn more about these campaigns as they evolve, please check out EMSL's website. Reach out to us if you have questions or ideas or thoughts. Thank you all for listening, and thanks to to Heather and others for organizing today.
This concludes the open to the entire public session for the critical minerals campaign meeting. So thank you all for attending. We appreciate your time you spent with us.